Good afternoon, everyone. My name is Michael Schatz. I'm an associate professor at Johns Hopkins University. Before I was at Johns Hopkins, I was at Cold Spring Harbor Labs. Before that, I was at University of Maryland. Before that, I was at the Institute for Genomic Research, Tiger. So I've been in genomics for about 15, actually coming on 20 years now. And it's just been really exciting over the last 2 decades just to watch this field explode. And a lot of this has been driven by kind of all the new technologies that have been developed in this time frame. So what I'm going to do this afternoon is tell you a little bit about some of these new technologies, about how they can be applied, some of the algorithms that can be used. It's going to be kind of sort of a survey of the methods. A few times we'll go a little bit deeper, but I just really want to kind of give a gentle introduction to the field to all of you. The talk today is split into kind of about 3 parts. The first part should be a very gentle introduction. I saw I was scheduled to come on right after lunch, so I just wanted to kind of give a little warm-up. And there's this little analogy I like to tell to kind of highlight some of the key problems in genome assembly before we talk about real data. In the middle, we're going to talk about some of the practical issues about what does it take to create a good assembly, what are some of the technologies out there, some of the requirements. And then in the final part, I'm going to tell you a little bit about my current research activities. So it's going to be kind of a split between tutorial and research talk. So the analogy I like to tell starts like this. So imagine you're Charles Dickens back in the day, and you just finished writing A Tale of Two Cities, which begins, it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness, and on and on and on and on. So Dickens, just finished his masterpiece, he's very excited about it, and he wants to go get it printed and get it, so he can distribute it, become very famous, make his fortunes and everything. So here's the one stretch of the analogy. So instead of getting it printed on regular pages, he's going to get it printed on a long spool of paper. Right? So this is my hopefully totally transparent metaphor for long strands of DNA. So he goes to the copy shop, he gets it printed up, he gets his spools, and as he's walking out of the copy shop, oh no, he trips and it falls into the paper shredder. Oh no. So it goes through, and it gets cut up in a very sort of fancy way, which is that every fragment now has exactly 5 words on this. So he's like, oh no, what do I do? Right? It's in the shredder bag, it's been all mixed together. All the fragments from the first page, the last page, the middle, all mixed together. There's any sort of locality is totally lost, right? It's all just a big jumbled mess here. So he's like, oh no, I just finished this masterpiece. I want to reconstruct it. I want to be able to sell it. I want to put it back together. So again, hopefully my totally transparent metaphor for we want to sequence DNA. The technology does not yet exist today to read entire mammalian chromosomes, but we do have this thing where we can sequence fragments of it and then hopefully reconstruct it. So A Tale of Two Cities is about 140,000 words long. There's some good trivia for trivia night sometime. 140,000 words long. So if we have 5 copies of the book going in, 5 words per fragment, we're going to have about 140,000 fragments associated with it, all mixed together. Critically, because of this shredding process, some of the fragments we're going to see are going to be identical. Now sometimes they're going to be identical, not shown here. Sometimes they're going to be identical because the cuts just happen to perfectly line up. That's kind of to be expected. And then sometimes the fragments are going to be identical even though they came from like different pages, different parts of the book at the same time. So here I've colored 2 fragments in yellow that both have exactly the same words, of times it was the, and in blue 2 fragments that have the words it was the age of. So and here they come from different spots in the book, they just happen to have the exact same words on them. So in this shredder bag you want to put them together. A good idea, I'm not going to really go over it today, but a good idea is maybe lay them out in alphabetical order. This enables things like binary search. If you've ever thought about suffix arrays or any sort of string matching, sorting is a good thing to do. It just sort of kind of organizes the data, but it doesn't actually help us directly reconstruct the book. So the process that we're going to use, and this is the process that all of the early genome assemblers, so the original Tiger assembler, early FRAP, early human genome assemblers, I'll use what was called the greedy reconstruction. So the process for this is you're going to start, maybe you have some hint about where the book began, maybe you're just going to reach in randomly, maybe you want to do some coverage analysis, pick the fragment with the highest, lowest, there's different starting points. But the gist of it is we're going to reach into our bag of fragments, pull one out, and then the whole business of genome assembly is, well, what fragment comes next? What fragment comes next? So here, this first fragment is maybe small in the back, reads, it was the best of. So kind of very intuitively, we want to find some other fragment that is going to match up against this. So formally, this is called an overlap, where the end of one fragment matches the beginning of the other. So we want to reach in here, we want to find some other fragment that has an overlap, and there's lots of possibilities that are here. You could have a very thin overlap, overlapping by 1 character, 1 word. You could have a little bit thicker, a little bit thicker, a little bit thicker. In a greedy framework, generally, you want to find the longest possible overlap, with the most characters in common between them. So here we're gonna go through our sorted list. We're gonna try to find the fragment with the longest overlap, and aha! I just found this fragment was the best of times that has a 4-word overlap. So taking these two fragments, each have 5 words, lining them up, looking at overlaps, I've just reconstructed the first 6 words of the book. And kind of more generally, I wouldn't know it was the beginning or the end, but here we know because of the capital letter, it's at least the beginning of the sentence. So like good computer scientists like to do, once we have a little bit of an algorithm, well, we're just going to repeat. Let me just go through my list of fragments here and try to find the fragment that extends it again, and hopefully in this sort of maximal way. So now I've been able to reconstruct it was the best of times it, do it again, it was, do it again, do it again, do it again. So far things are going really well. As we go and sort of search through our list of fragments, something interesting happens on the next one, which is that there are going to be 2 fragments that both overlap equally well, but the fragments have the same words on them, so no problem. We know unambiguously it was the best of times, it was the. But as we go and we try to find the next fragment, something really interesting takes place. So we've walked through the reconstruction this far, we go to figure out, well, what's the next fragment in this book, and suddenly the fragments start to disagree with each other. So now we have, at the end here, we have 2 fragments, each 5 words long. One reads times it was the worst, and one reads times it was the age. So putting this all together, we've reached this sort of ambiguity, and we're not sure, there's just not enough information, at least not here, to be able to decide, should it be, it was the best of times, it was the worst, or it was the best of times, it was the age. Right? So this is kind of this is one of the central challenges of genome assembly, is recognizing these ambiguities and then deciding what to do. Why did we have this ambiguity? It's because of the repetitive sequences, right? We get these fragments that are identical. Even with perfect sequencing fidelity, we get these fragments that are identical. As you walk into them, no problem, but then as you walk out of them, you're just not sure where to go. So how are we gonna, we only put together a few words, right? Dickens is really frustrated. Oh no, I want to find the whole book. We have to think harder, smarter. We need a sort of more global view on how to do this reconstruction. Throughout the years, a variety of approaches have been proposed. Most of them use one of a couple different graph formulations to solve this problem. The one I'm going to sort of walk through now has a funny name. It's this de Bruijn graph construction. This comes up quite a lot, especially for using short-read sequencing data. It has some really nice properties, and is a very popular approach for this book reconstruction, genome reconstruction problem. So the way that it works is quite unintuitive, at least at first, which is we're going to take short fragments, and the unintuitive thing is we're going to make them even shorter. So in here, we're going to start with 5-word fragments, and we're going to go down to even shorter 4-word fragments. In sort of real, genomics data, this would be, 100 base pair Illumina read. Maybe we chop out the first 25 bases, shift over by 1, the next 25 bases, the next 25 bases, the next 25 bases, until you've tiled across the entire read. Critically, though, as we're doing this sort of substring operation, we're going to keep track of the order to those subfragments. Remember, okay, you're the first one, you're the next one, you're the next one, you're the next one, you're the next one. And then we're just going to keep track of those as little directed edges in our graph. And then critically, whenever we see the same substring, even if they come from different fragments, we're going to keep track of all of those directed edges so that we can build up a graph. So here I have 2 fragments that both start with 5 words. We chop them up into 4-word subfragments, keep track of the directed edges. That gives us this little beginning of the graph reconstruction here. So there's a long history to this. It was first proposed in a purely mathematical context back in the 1940s as a way to kind of enumerate all possible strings given some alphabet. And then it's become, critically, I would say, in the 90s and early 2000s, really central in genome analysis. So why do we do this? Let me kind of prove by picture why this is a good idea. So this is going to be me sort of simulating Dickens, taking that list of fragments, from each of those 5-word fragments, deriving 4-word subfragments, keeping track of the edges between who's who, and then aha! It's tedious for people to do. It's really easy for computers to do this work. And then if you just sort of lay it out carefully, suddenly the entire book starts to emerge just from this graph, just from these relationships between the fragments. So here, by construction, every node has exactly the same length. Every node here is exactly 4 words long. In the original de Bruijn graph in a genome assembler, all of the initial nodes would be, say, exactly 25 characters long, something like that. A very natural thing you're probably saying to yourself is, oh, Mike, certain parts of this graph are very easy. where you have one node followed by exactly one, by one, by one. Let's just go ahead and just sort of find those regions and let's compact them down. Let's compress them down. And indeed, that's exactly what basically every genome assembler will try to do, is once you've built this initial graph looking at short, exact, fixed length words, we're going to try to extend this. This is sometimes called compression, compaction, some sort of simplification of this graph. And then, aha, suddenly now we've reconstructed bigger parts of the text, bigger parts of the genome through this quite simple process. So now we have, I don't know, 6 or 8 nodes that are left, even though we all started from those fixed length sequences. Now you might be tempted to be like, well, Mike, it almost certainly starts in the top left, and oh, Mike, you have this one sort of dangling edge. That's probably where the rest of the book is. Let's try to find some tour through this graph that is going to visit every edge, and that should probably reconstruct the book. And indeed, in this very, very simple example, that would get you pretty far. And that's because in English, it would be really, really awkward to spell out, it was the best of times, it was the worst of times, it was the worst of times, it was the worst of times, it was the age of wisdom, it'd be very, very awkward to do that. However, this graph is fully compatible with such a reconstruction. So if we're going to do any sort of analysis, we have to be really careful about how many times we're going to visit every node. And furthermore, this is just like the very beginning of the first sentence of the book. If you kind of zoom out and you take all of the words in the book, for whatever reason, Dickens sort of liked this sentence structure. You get these more tangled, nested graphs where there's many, many possible paths through there. Even though it's constructed in a very simple way, the graph that is derived can be very complicated. And you might step back and say, in CS 101 or 102, we learned about how you can find Eulerian tours in linear time. This is an easy problem. But again, the problem isn't so much finding a possible assembly. Like, so here, let's imagine we're going to visit every node, a minimal number of times. It could go blue, red, yellow, red, purple, green, or it could go blue, red, purple, red, yellow, red, green. Even in this little simple motif, there are 2 possibilities. They're exactly ambiguous. You have no idea which is which if this is all the information you have. So I call this my little clover leaf motif. If I had a clover leaf connected to another clover leaf connected to another clover leaf, suddenly you get this sort of combinatorial explosion of how many possible graphs can be derived. In fact, we did some analysis a few years ago where we were looking at every microbial genome that was available at the time and sort of considering, let's imagine we had infinite coverage of short sequencing data, and then we built this graph out of 25 mers. The number of possible tours to this graph was much greater than the number of atoms in the universe. It's just like one of these enormous problems. Normally I would call these an astronomically hard problem, but because it's so much more difficult, we started calling these genomically hard problems. So it could be another piece of trivia to take back with you. We have the hardest problems in the universe. So we can, in linear time, we can find such tours, but we need some way to evaluate what's good, what's bad. Other sort of formulations for genome assembly are almost always NP complete or NP hard. So inevitably we're going to have to fall back to certain approximations, certain heuristics. And then critically, we can try to bring in other data so that maybe we can resolve the order of these little nodes so that we can resolve some of these clover leaves. In lacking of other data, or maybe you don't have quite the right other data to be able to assemble the whole book or the whole genome, another that will happen is you'll run your assembler, you're going to get a set of contigs. So a contig is a contiguously assembled piece of sequence. You're going to get your contigs. Some of these contigs will be very big, some will be kind of middle, some will be small. And basically every project I've ever run, you get this long tail of many, many, many small contigs. What happens is you get any sort of repetitive sequences are hard to resolve. So you just get, the assembler will output these little pieces. Now, given a size distribution like this, if you take like the mean or maybe even the median, because there's this long tail of small contigs that'll tend to sort of dominate the size distribution, it'll seem like you don't have a very good assembly. In response to that, for decades now, people have been using this N50 metric when they talk about assembly contiguity. So an N50 number is like a weighted median. It says that 50% of your genome has been assembled in the contigs this size or larger. It's like a median. And it's very straightforward to compute. You just take your contigs, you lay them out from largest to smallest, you kind of walk through there until you get to the halfway point. In assembly A, the halfway point is a contig that is 30 kb long. In assembly B, the halfway point is a contig that's only 3 kb long. So you'd say, well, the contiguity of assembly A is about 10 times that of assembly B. If you have to pick sort of one number to describe your size distribution, this is the one that has sort of become popular in the field. Of course, if you knew the rest of the distribution, that would be good too, because you could have funny things at the halfway point that are not representative. But in general, something like this is what you're going to see in almost every project. So if you read any sort of genome paper someplace, for a while it was buried in the supplement, then it was in the abstract, and now unfortunately it's kind of getting buried in the supplement again. Someplace though in the paper, it'll talk about this N50 size as a way to kind of show off in a single number, how good your assembly is. In sort of all things being equal, bigger is better, right? So if we get bigger N50, that means our contigs are better. That means if we're going to go, I don't know, look for genes, flanking regulatory sequences, our chances of finding them are going to be improved if we get bigger chunks. If we're looking at sort of complex structural variations like we heard about this morning, having bigger chunks is good, so we can sort of isolate this. If you're doing, I don't know, looking at synteny of chromosomes across evolutionary time, you need big chunks so you can map these out. So N50 is good. However, you got to be careful, right? Because now that this is our metric of success, there is a temptation, or maybe not even a temptation. There's just sort of a danger, that you could be sort of doing bad things to smash contigs together, right? So if you remember, contig is built out of these overlaps. Now let's imagine we're going to do something crazy like accept one base pair overlaps, right? So suddenly every contig will overlap 1/4 of all the other contigs. You can start to do crazy things and get these artificially inflated sequences where horrible things will happen, where part of chromosome 1 gets assembled with chromosome 2, gets assembled with chromosome 15. Crazy things can happen. But in general, all things being equal, you hope a bigger N50 is reflective of better contiguity. Okay, so hopefully that was my simple introduction. We're going to take our sequences into the Bruijn graph. We're going to chop them up into short sequences, build up a graph to capture those relationships, walk through the graph as best we can, and get some contigs out. Okay, so moving right along. So what are some of the sort of more nuts and bolts about this in genome sciences? So the first thing to recognize is that assembly is a pretty ubiquitous problem, right? Anytime, throughout this meeting, we've heard about, we looked at this genome, we looked at this genome. The instruments do not give you chromosomes. They don't give you genomes, right? They give you short fragments. So almost every problem in genomics is sort of enhanced or maybe is really central to this genome assembly problem. And I know many of us are studying human genomes, so we've had the benefit of literally billions of dollars of investment to build up that reference genome that we can all leverage. But as you move into beyond human, into different species, or perhaps even very sort of mutated versions of genomes where we have a reference. We want to be able to do this, solve this assembly problem de novo, where we're just going to collect this sequencing data, reconstruct the genome. A very sort of a great example of how this is being used today is there's something called the Genome 10K, sort of being morphed into this thing called the Vertebrate Genomes Project. And I'm teasing, but they sort of went to the San Diego Zoo and went, yep, we want to sequence everything here. The idea being is once we've done all these assemblies, we can do phylogenetic analysis, and we can basically build a time machine to look over any sort of evolutionary time that we might be interested, be able to look at how sequences have been mutating, how they've been conserved. It's an enormously important project to be able to catalog all these variants. Even within sort of a more focused clade, there's something called the Insect 5K project. Sequence 5,000 insects. I just heard about the Bat 1K project to sequence all bats on the planet. So basically every sort of major species, every sort of family that has any sort of interest, there's interest now to do assemblies of those. Another sort of application of this is not exactly the same problem, although it's definitely going to leverage a lot of the same algorithmic ideas, is this so-called problem of metagenomics, where we're not going to try to isolate one sample at a time, but we're going to sort of extract DNA from a complicated sample. So this could be, I don't know, all the microbes living inside of you, on side of you. This could be microbes living in different environments and so forth. A great example of this is the Human Microbiome Project, where they've been doing exactly that, sequencing all the microbes from different body sites, learning a lot about what does it mean to be healthy, what does it mean to be in a disease state in terms of the microbial composition there. If I had to give sort of one anecdote as to why that might be interesting, there's been a set of papers looking at obesity in mice. And one of the observation is if you take skinny mice and you take obese mice and you swap their stomach contents in a very gross way, the obese mice become skinny and the skinny mice become obese. So, very suggesting that these microbes played an enormous role in our metabolism. Another example comes from the Global Ocean Survey led by Craig Venter and friends, where they were sailing around the world, visiting all of the world's most beautiful beaches, and along the way collecting seawater, which seems kind of like a crazy project. But I find this highly analogous to like the sky surveys, where they're just surveying all the galaxies, all the stars out there. So what did they discover as they were sampling all the seawater? Well, they expanded the list of known genes by something like a factor of 100. Just by all the diversity that were out there at the time. So it's just, there's amazing diversity we've just never seen before, and it's just kind of all around us. Another important application for these assembly technologies is, maybe you're not really interested in, a new genome, but maybe you're interested in structural variations. We heard the nice talk this morning. Or maybe you're, something like, oh, I'm interested in the transcripts or other sorts of, sequences where, we need to be able to, we can only get a partial readout. We need to put it together. We're going to use these assembly ideas to do so. The process for all of this is just like we saw with Dickens, right? We're going to take our sample. We're going to shear the DNA, sequence it, and then from those relatively short sequences relative to the chromosome length, we're going to assemble some sort of, build up some sort of graph from that data. This could be the Bruijn graph that I just showed you. Another popular graph all starts with overlaps. So especially as we're looking at long read technology where we have, a 10,000 base pair read and another 10,000 base pair read, it's a little bit crazy to chop up 10,000 base pair reads into 25 mers. So let's look more directly to compare those reads to each other, try to find those long overlaps. That'll build you a graph of, read 1 overlaps read 2, overlaps read 3, overlaps read 4. In the same way that we can simplify the Bruijn graph, we try to simplify this graph. That'll give you an initial set of contigs, and then we have to bring in some sort of, either we're done, or we try to bring in some sort of other data, so mate pairs, optical mapping, Hi-C, 10x genomics, linked reads, some other data to be able to assemble entire chromosomes. If you want to do an assembly, there are certain ingredients that you just have to have. Otherwise, your assembly project is doomed. The number one on this list is coverage, right? We want to be able to put together the whole chromosomes. If I haven't sequenced every base of the chromosome, it's hopeless to be able to reconstruct it. We'll talk a bit more about that. Another really important is sufficient read length, right? So we're going to be building up these graphs. Here it's totally ambiguous if green is connected to yellow or green is connected to purple. But if we can overlay some sort of information, this is that scaffolding problem. If we can bring in some other information, we can say, aha, green is connected to yellow through that red repeat. Blue is connected to pink through that red repeat as well. And then the final sort of ingredient is you need high quality data. And here I mean very broadly. So if there's sequencing error, we want the consensus to be accurate. We have to accommodate for this. The worst project, or the most doomed project I ever heard of was, for whatever reason, they were sequencing this tree where they had grafted on branches, and they thought they were collecting DNA from one part of the tree, and it turned out to be a different part of the tree, and it was like a totally different genome. So, when I mean quality, I mean from sample collection, library preparation, sequencing. You really, and hopefully no software bugs. You need, to get a high quality result, you need high quality ingredients going in. So of these, the first is this coverage problem, and I think a lot of this will be familiar to a lot of you. It really begs the question of how much coverage do I need, right? So the analogy that I often think about is, maybe not here, but in places where it rains, if you start with a totally dry sidewalk to start, right? The first raindrop falls, it's like a pinpoint. It'll land on the sidewalk. It only covers a tiny, tiny little bit of the area. The next raindrop falls, another little pinpoint of area. The next raindrop falls, another pinpoint of area. Eventually, though, you hope the raindrops will touch. That'll grow these little puddles. And then eventually, even though the raindrops are teeny tiny, you can saturate the whole surface, right? So by oversampling enough, you can saturate the whole surface. You can get a readout of the whole genome, even though individual reads are more or less randomly distributed across the chromosome. So the question is, well, how much sequencing do I need to do? How much rain do I need to collect in order to saturate an entire sidewalk? And the caveat is something like, well, every raindrop costs you $1. Maybe that is true here in California. Every raindrop costs you $1. So you have a limited budget. You just can't sequence infinitely. So sort of critically, make it really concrete. If the genome is 100 million base pairs, should we sequence 1 million 100 base pair reads? That will give you a total of 100 million bases. Do we think this is a good idea or a bad idea? What? I'm seeing some thumbs down. I mean, it's 100 million bases. I collected 100 million bases. This seems like good. Okay, I can't fool you guys, right? So no, right? If you want to sequence 100 million base pair, I don't know, C. elegans genome, you need a lot more than 100 million bases of data going in order to cover it. And it's all because of these statistical properties. So here's me sort of simulating a genome in silico, a little bit of R, where I'm just going to be taking raindrops and then just letting them fall randomly. In this example, let's imagine that it's sort of a number line, labeled 1 to 1,000, and I'm going to let 1,000 drops of rain. So on average, every position should get 1 raindrop, but there's going to be a distribution. Some places will get lucky, and multiple raindrops will fall there. However, though, because some places get lucky, other places get unlucky, where there's no raindrops falling in there. So this sort of profile at the bottom is that coverage that are there at the individual raindrop level. If I tilt that on its side, build a histogram, you get a plot like this, maybe hard to read. But kind of the punchline from this is if you've done 1,000 raindrops into 1,000 buckets, about 35% of the buckets will be empty. This is just one of these statistical facts. Okay, we can live with that. We go collect some more money, do some more sequencing. We throw another 1,000 raindrops in here. So now on average, there should be about 2 raindrops per bucket. Some of them are lucky, some of them are unlucky. But still, we're left with about 15% of the buckets that are empty. We can double the coverage again. Now we're down to just a few percent of the buckets that are empty. We double the coverage again, and now less than 1% of the buckets are empty. So for a long time, this rule of thumb about 8x coverage is the minimum is entirely driven by this sort of analysis about if you do less than 8x coverage, you will for sure be missing at least 1% of your genome. 1% of 3 billion is a big number of bases to be missing in the case of the human genome. Does anybody recognize this distribution? Hopefully many of you do. Anybody? It's okay. I hear a lot of whispering. So, okay. Well, this part at 8x, that looks like a normal thing, right? But if you look at lower coverage, it doesn't look so normal, right? It goes out to 6 or 7. It doesn't make sense to have minus 7x coverage. So it's definitely not normal on the low end. Does anybody know what this is called? Okay, well, I think we need to go back to Stats 101, right? So this is a really concrete example of where the Poisson distribution can come from, right? You just distribute balls in the buckets, you count how many balls are in each bucket. That's the Poisson distribution. That's why earlier today we heard about, we model this as a Poisson. That's because in the limit, if the sequencing is totally random, totally unbiased, we expect something like a Poisson distribution in coverage just from the sampling that takes place there. A normal distribution has 2 parameters, right? It has a mean and it has a standard deviation. Poisson is kind of special because it just has 1 parameter. It just gives you a mean. And then the key, more trivia for tonight is the standard deviation will be the square root of the mean. So okay, let's go ahead and impress all your friends. I'm sequencing C. elegans. I have 100x coverage. I know that there's going to be some fluctuation. Sometimes I'll have a bit more. Sometimes I'll have a bit less. What would be the 2 standard deviation range that you expect at 100x coverage? Oh, come on. Standard deviation on 100x in a Poisson. The square root of 100 is 10. Okay, I know it's right after lunch. So we expect there to be about 80x coverage to 120x coverage, right? Just based on that Poisson distribution having sort of a fixed parameter there. I should caution, in real data, the data are often much broader than that, right? A Poisson is actually a very strong statement about how tight the distribution is. Often with real genomic sequencing, the distribution is much broader. And that's because there are these subtle biases. This morning we heard about GC bias. That will make certain regions undercovered, which means other regions will be overcovered, which will broaden the distribution. That's why with real data, you probably want to use something like a negative binomial, which gives you a free parameter to adjust for that over dispersion. Okay, so we've done our sequencing. We've let the raindrops fall. A natural question is, well, in some species we have a pretty good idea of the genome size, but if you're branching out in a totally new genome, you might not have a very good estimate. The worst project I ever worked on, they swore up and down it was a 20 megabase pair genome. We get the data, no way this is a 20 megabase pair genome. It's like a 200 megabase pair genome, off by an order of magnitude as you do this analysis. A good way to sort of demonstrate this, especially with Illumina sequencing, is scan through your data and just count all the Kmers. Cut out the first 25 characters, make a table. How often do I see this? You might have to do something fancy because we're going to be sequencing the forward and reverse strands, but no problem. There's lots of great tools, jellyfish and so forth, that can do this analysis, even for large data sets, very, very quickly. So we can just build up this histogram. This is sometimes called the Kmer profile. It says how many Kmers occur once, twice, 3 times, 4 times, 50 times, 100 times. And what you're delighted when you see something like this, right? You get this nice peak, sort of Poisson shape, not too broad, centered in this example at about 40-something X coverage. That's good, right? That means even if I didn't know how big the genome was, I'm pretty confident there I have about 40X coverage. There's some caveats to this. This is 40X Kmer coverage, not base coverage. There's a correction factor, but we know how to deal with that. On the left there, very close to the axis there, you're often, in every project I've ever seen, you're also going to get a secondary distribution. These are going to be Kmers that occur once, twice, 3 times. What do we think that those Kmers are? Yeah, here's some people. Sequencing error, right? So if I have 100 base pair Illumina read with a sequencing error rate of about, say, 1%, we expect on average there to be 1 mistake in every Illumina read. We don't know, it gets concentrated in the end, although we don't really know that until we've looked at it. It's very unlikely, assuming that your Kmer length is long enough, that if I just flip an arbitrary base, is that going to get me to another position in the genome? Especially as you're looking at 25 mers or longer, and even in the case of the human genome, more than 90% of the genome is unique at that level. So it's very unlikely that that will get you someplace else in the genome, so this is going to create a novel Kmer. And if it's that sequencing error is just random, it'll just be in that one read. Kmer will look weird, and that it won't be supported by any other Kmers. Me and others have sort of noted this, and then you can write an algorithm that will go identify these error Kmers, go back to the read, and then just, it's a little bit more sophisticated than this. We can just think about like systematically permuting every base in the Kmer that it looks weird until you find it, a substitution that will take you to some other Kmer that's well supported. So this is the statistical error correction, which is going to identify what is likely to be errors, flip the bases until I've created a new Kmer, a new read that is well supported. Hopefully you've done a good job, and that is, just doing error correction, not error corruption. That's the danger there. Another fancy thing you can do there is you can look at the sort of peaks that are there. You can do some statistical modeling to infer a lot of genome properties, even if you have no idea what the true, what the true genome size is. Just by looking at this Kmer distribution, you can look at the total genome size, how repetitive it is, if it's heterozygous. You can look at your effective coverage, PCR duplications. Lots of great properties can be derived just from that Kmer spectrum that is just counting the Kmers, I was thinking about presenting that, but there's other better stuff I want to talk about, but go check out the paper to learn more about that. Okay, so we have our genome, we've done our sequencing, we've confirmed that we have good coverage, we've done maybe some error correction, or maybe let's imagine we have perfect data. We're going to build up the initial graph, we're going to simplify it. My chromosomes are supposed to be 100 million plus characters long, and my contigs are, in the case of Illumina sequencing, 10 kb long. So why are these contigs ending? Why are these contigs ending? There's actually several right answers to this question. Yeah, so as we heard in Dickens, one of the central challenges are repeats. And even this little example, that red segment in the middle is probably repetitive, has multiple ways in, multiple ways out. So that's one of the right answers. What's another right answer? Heterozygosity, another great answer. Maybe the error correction procedure that we tried wasn't 100% effective. There's a little bit of residual error. Or maybe, as much as we tried to oversample the genome so that we thought we'd get every base, maybe this green contig should have been connected to another contig. We just lacked coverage. So those are kind of the top 4 reasons, why these contigs might terminate. In my mind, the number one challenge, though, is repeats, right? Because they're ubiquitous. They're just really fundamental. Even with infinite coverage of error-free data, they're going to be challenging. And furthermore, in mammalian genomes, it's bad news because it depends on how you count and everything, but about 50% of mammalian genomes are repetitive, consisting of complicated stuff ranging from microsatellites, line and sign elements, LTRs, transposable elements, others just sort of complexity there. A useful metaphor when you think about the genome is a graveyard for ancient transposable elements that have been jumping around making copies of themselves, making copies of themselves, making copies of themselves, until they acquired mutations to the point where they can't jump around. So this ancient graveyard is kind of my mental model when I'm thinking about any new genome. That's mammalian genomes. As you look at some of these enormous plant genomes, things tend to be even worse, and that these transposable elements just get unleashed, and you just get them accumulating in huge numbers very, very quickly. Good news is, from the point of view of the assembler, as it's looking through this, if this is my sort of cartoon genome, which has unique segments ABC, repetitive sequences in red there, from the point of view of the assembler, it's going to sort of bring together all those reads from the repeats, and there's some pretty easy statistical, assuming that the data are not too biased, there's some pretty easy statistical analyses you can do to recognize where the repeats are, again, with the assumption that there's a Poisson sort of coverage that you expect there. So once we have that, we can build our initial contigs out of real data. Then we're going to try to bring in other data, hopefully bring in other data, so that we can go from these relatively short contigs, and then figure out the relative order and orientation. Again, this could be mate pairs, linked reads, high-C optical maps, genetic maps. Here, maybe we have some mate pair data, so we know that A and B should be much closer than A and C. That'll help us, determine the order. That'll help us hopefully get entire chromosomes. As we do this with real genomes, it's likely there are still going to be parts that are going to be too complicated. That's why when even today, when you open up the reference human genome, it's like NNNNNNNNNNNNNNN. It's because from this marker information, this linking information, we knew that this contig and this contig should be a certain distance apart, but we don't know what that sequence is in the middle, even with all the technology that we have in the world. So we can fill those in with ends. That's why those ends still persist. So assembly quality, coverage, 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 coverage. By the way, if you're doing anything in genomics, coverage, 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 coverage. If you have low coverage, your project is doomed. So go out and make sure you have the right resources that you need. Number 2 challenge is repeats, 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 repeats. Oh, it's a self-fulfilling prophecy. Notice though that repeats is really dependent on your read length. If there was a magical technology that could read off 100 million bases in a row, no repeats. We're in really good shape. We're not quite there yet, but although it's been pretty exciting in the last few years. Error, surprisingly, is the easiest problem to deal with. Given today's technology, if it was like 90% error, that would be challenging. But with Illumina quality, for sure, we know how to handle that. Even at 10%, 20% error for some of the other technologies, we have a pretty good handle on that. And then the actual process is very hierarchical. We're going to start from individual reads, do some sort of overlap comparison, build up our initial contigs, and then hopefully build up our whole chromosomes. Okay. So now moving on, give you just 10 minutes on kind of current research. So current research, the good news is we have a lot of new technologies at our disposal, from sample preparation, enhanced linked read sequencing, enhanced long read sequencing, all kinds of cool technologies for chromosome mapping. Of these, for de novo assembly in particular, of these that I'm most excited about is single molecule sequencing. Right, so on that blue thing there is a histogram of read lengths. This is actually a little bit old now. But instead of Illumina reads that are all 100 or 200 bases long, I basically throw away every read that is less than 10,000 base pairs long, and I just measure that in 10,000, 20,000, 30, 50. This is a picture of the PacBio RS2. The longest read I've ever seen from that was a bit over 100,000 base pairs. Some of the new technologies we'll get to in a minute are producing reads upwards of 1 million bases in a row. If you're doing any sort of assembly work, getting 1 million bases in a row is great. Because if there's any repeats there, you don't need to figure out this complicated graph. You just read right through it. The big challenge for both PacBio Oxford Nanopore is that they have sort of miserable base fidelity, and that there's about a 10-ish% error rate depending on the reads and the time of day. And it's all dominated by indels. In the case of PacBio, it's dominated by phantom insertions, where sort of nucleotides float into the sequencing chamber. You see them, but they actually aren't being incorporated on the template. In the case of Oxford Nanopore, it's all dominated by deletions, where you're doing your analysis and you just sort of miss bases along the way. You skip them. So, kind of metaphorically, these are great for genomics because you get these huge chunks of the chromosomes, but they're very fuzzy. They're going to have 10% error or more associated with them. You have to make sense of them. The good news is to do assembly, to do anything in genomics, we're going to be oversampling. So we're not just going to get one readout. We're going to get one read with 10% error or 15% error, another read, another read, another read. And through some modeling and some real analysis, you can see that the sort of consensus accuracy improves quite dramatically with coverage. So here, there's a little bit of sort of even and odd, which is why it goes up and down. But overall, it drops exponentially with the coverage to the point where even though you start with, say, 20% error, at, say, 20-fold coverage, you're going to have less than 1% error remaining if you can get them all sort of lined up correctly on top of each other. In fact, with real data, we tried to measure this. I was one of the authors of the FALCON-unzip paper, where we wrote an assembler, sort of leverage this idea, where we get many of these long reads stacked on top of each other. We take the consensus. That becomes the final Contig consensus. We measure this very carefully, as carefully as we possibly could, by comparing to hand-polished Sanger sequencing data, and we determined the accuracy to be about 99.99% given enough coverage. It is true there is still errors. In the case of PacBio, it is highly concentrated in homopolymers. They are still out there, but 99.99 is pretty good. So the analogy is big fuzzy pictures to start, great algorithms, great software, quantitative scientists to the rescue. We can correct that. We can put eyeglasses on that so we can see that. Given that sort of technology, we're now getting beautiful assemblies across the tree of life. We're getting essentially complete microbial genomes, yeast genomes, even higher eukaryotes, fish species, plant species, human genomes. This is old now. We're getting these beautiful assemblies that come right out of them. One of the most important applications in my mind that I find to be really meaningful is now we're starting to apply this to cancer data sets. And the reason for that is genome instability is one of the hallmarks of cancer, where the genomes can get scrambled, the chromosomes can get fused together, they can get rearranged to the point where it doesn't really look so much like the reference genome. Maybe we need to do some de novo sequencing, de novo assembly, so we can get a better look at what is there. To get started, we sequenced a breast cancer cell line called SKBR3 with PacBio long-read sequencing. We generated a total of 75x coverage. Our mean read length was about 10,000 base pairs. Our longest read was about 100,000 base pairs. I should highlight, this was really Maria Nadestad, who is a former PhD student of mine. This is her thesis work. She's now back at DNA Nexus doing great work with their visualization team. Based on all the great stuff we heard this morning, we were really interested in structural variations in this cancer genome. We used both an assembly-based approach where we assemble with Falcon. We align those contigs to the reference. We sort of carefully look at the breakpoints in that way. From that, we can detect about 11,000 structural variations. We also did a split read approach where we align the reads with our new aligner NGMLR. We sort of scan through those alignments using a tool called Sniffles, and there we could find about 20,000 structural variations. The big limitation with the assembly is because it was so complicated, where there was lots of aneuploidy and rearrangements, the assembly graph gets really confused, so you don't get as sort of clean a resolution as you would hope. With the split read alignments, though, if you use an older aligner like BWA-MEM, it does a bad job. If you use a new aligner like NGMLR, it does a much better job. The main reason for this is these long reads are filled with indels, right, as I mentioned. And most kind of older aligners use something called an affine gap penalty, where the indels are scored in a particular way. But if you have small indels from sequencing error, that confuses when you have big indels from real structural variations. So you need kind of a dual scoring mode. The approach that we use for this is called convex scoring. Actually, it's kind of a heuristic based on convex scoring. Please check out the paper or come talk to me if you have more questions about that. The punchline of it, though, is given the right scoring model, even though there's 10% indels, we can get really nice alignments of these data, and then the structural variations are clear as day. This is the same data aligned to what should be a homozygous deletion. With the new aligner, you get really clear indications of the deletions. With the old aligners, you get a miserable resolution. You might think it's heterozygous or something. So now we have really accurate variant calling in that way. This is a Circos map of all the variants that we could find. Let me just, since I'm out of time, let me kind of jump to the headlines. We're finding tens of thousands of additional structural variations. We did PCR validation over a large sampling of these, confirms the really high accuracy there. We detected many novel gene fusions. We could even sort of reconstruct the history of the cancer to identify early and late mutations in the cancer. So this was just published in Genome Research, and knock on wood, it'll be hopefully the cover story of that journal in August. So that's all old. Let me just give you 5 slides, if you can bear with me, on some of the new stuff. So this was all done with PacBio sequencing. We got started a couple years ago. It was a long process to sequence. We had to write all kinds of new software to make sense of it all. If you were to redo this project today, it would still be expensive. It would be, I don't know, $30,000 or $40,000 worth of PacBio sequencing, to the point where it's not really possible to apply this to many more samples. We did it once, hoping the cost would come down. They have. But with PacBio, it's still not there yet. As a result of that, we've become super interested in nanopore sequencing, and I suspect a lot of you are. So the nanopore, I'll just give you 2 slides on this. So nanopore sequencing, This is a sequencer. I don't know who else flew with a sequencer in their bag, but I did. Unfortunately, I didn't bring any flow cells. Otherwise, we could go sequence at the beach tonight. Maybe next year. So it's a thumb-sized device powered over USB. Inside there is an array of sensors. Inside those sensors, if you zoom in, there's a membrane that establishes the top and bottom half. There's a protein pore that pierces through there that allows ions to flow. It's just like electrical current can flow through there. And then the gist of it is whenever there's a new molecule that is flowing through that pore, that'll disrupt the current that is there. And hopefully, there'll be a recognizable signal, right? So this little, yellow, red sphere, whatever that is, will give you one change of the current. Other sort of molecules will give you a different change to the current. So the way we use this for DNA sequencing is there's like a half a day prep or less, half a day prep to prepare the molecules. And then there's what's called a motor protein will lock onto double-stranded DNA. It'll unwind the two strands, so you get single-stranded DNA passing through the pore. There's an electrical readout that is taking place as the molecules get ratcheted through. I should highlight that the sort of relevant time spans are, you might get many seconds to minutes of sequencing, but the changes to current are measured in picoamps. This is like the most exquisite current meter you can ever imagine to be able to recognize that. From there, there's a signal processing pipeline where you go from that raw signal data of that raw current data. It gets segmented into events. In the old days, we used an HMM type approach to then be able to recognize, well, what was the most likely nucleotide sequencing that was compatible with the sequence of events that we saw. It's all quickly moving to recurrent neural nets. You just get much better resolution with that. Another thing that you may not be aware of is this is sort of the baby instrument. In fact, this is my son James 2 years ago posing with his favorite rattle. It's about a $1,000 device, but a whole human genome to 50x coverage would be something like $15,000. So, it's great in that you get long reads. The longest read I've ever seen has been about 1 million bases. Other people are reporting reads a little bit over 2 million bases, but it's relatively costly to do so. The daddy of this device, here's me posing with it, is called the Promethean. It's like a desktop instrument with much greater capacity. It's more expensive to get started, but has much greater capacity. Also, it has much less expensive sequencing costs. So now getting 50x coverage in reads averaging, say, 10,000 base pairs or maybe 20,000 base pairs is like a couple $1,000. So it is more expensive than Illumina sequencing, but not that much more. And the read length is, 1,000 times greater. So there's lots of exciting opportunities for it. We've been using this, so the breast cancer work I presented that's now published was all done on a cell line, but now we're starting to be able to take it in the clinic to be able to look, in patients, to be able to see what variants are there. We've tried this with, directly with their own tissue. That works okay. It depends on how heterozygous it is. It also depends on how big the tumor is, these sorts of things. So for some of the samples, we've been deriving patient-derived organoids. So I don't know how familiar people are with organoids. This was it's sort of like a cell line. It's sort of like a cell culture, but it's done in 3D. You get these little spheres of balls that are living cells that you can then go treat. You can go do RNA-seq. They're like little mini organs, hence the name organoid. It's a very sort of complicated bench procedure to develop these, but then once you have them, you have this very stable set of cells that you can go profile. Critically, these are great for long-read sequencing because it's a great way where you can establish really stable collections to derive high molecular weight DNA. We've been starting to do this. I'm out of time, so I'll just give you the punchline. It remains challenging to get high molecular weight DNA, like 100,000 base pair segments or longer, but we're making great progress there. Really, the transformative thing was when the Promethean started working, and now we're starting to get north of 70 gigabases on a single flow cell, which just changes the world, right? This is approximately 10 times as much data as you'd get off of a PacBio SQL flow cell for similar costs. It's just really transformative. We sequenced both the normal tissue and the tumor. The thing that we knew to go look for was that we were going to see thousands and thousands of structural variants that were specific to the tumor. The really unexpected and exciting thing is we're also seeing interesting things in the normal tissue. And here's an example of one, which is a repeat expansion in BRCA1 that was only detectable in the Nanopore sequencing. This poor patient, had a breast cancer panel, detected nothing, had short read sequencing, detected nothing. It's only through the long read sequencing that we can see these variants start to pop up. So just to sort of summarize and wrap up, right? So there are these amazing new technologies that are out there. We're starting to see things that have never been seen before. In the case of, say, breast cancer, maybe not all, but maybe in some cases we know what the right genes are. We just haven't been using the right technologies to go look at them. So it's really up to us and you to write the software that can make sense of these data to detect such variants. Last night we were kind of chit-chatting about what we thought would be good problems. So I would strongly recommend you avoid very narrow problems like new Illumina or maybe even PacBio-based callers. Just too narrow. Writing an entirely new genome assembler from scratch I think is too ambitious. The good news is most assemblers like GNU are highly modular. So if you're interested in assembly, focus on one piece. You're not pointing in the right direction. A good problem to work on would be any sort of methods that are robust to errors, polyploidy, aneuploidy, complexity in the sample. I think that's our new reality, is now that the reference genome is really established, the simple problems are kind of really well established. We need to go beyond that. Another really good problem in my mind is if, for the foreseeable future, we're going to have a few samples that have long read data and then like 1 million more that have short read data. Is there some way we can use the long reads to inform the analysis of the short reads? I think that's a really good problem. And then some of the best problems are if you can synthesize, huge number of samples, many different data types, that's going to be a great thing so that we can prioritize some of the variants. If you want to hear more, me and some colleagues, we wrote a little review to walk through these issues. Lots of great people help with this. And then very finally, I'm co-organizing a conference later this year. We're going to be talking a lot more about these methods. Come join us. Thank you very much.
So we have time for one question maybe?
Yeah, sorry for going over.
Any question?
So you talked a little bit about N50, right?
Yeah.
Like, that doesn't say anything about accuracy.
Correct.
Can you say something about like how do you evaluate accuracy of assemblies?
Yeah, great question. So this N50 is one summary statistic of your size distribution. Tells you nothing in itself about accuracy. What is going on is, it's hard to assess accuracy if all you have is sequencing data. One thing you can do is you can basically realign your sequencing data to your assembly and look for consistency, right? If you have spots in your assembly where the coverage drops to, it should be 100x and then it suddenly drops to like 1x or maybe even 0x, you would say, how could you get 0x coverage, but sometimes assemblers do? That can be a good clue that there was a mistake there, although it could be, could be legitimate. In addition, if you can bring in orthogonal technologies, that can be a really good way to assess where there's errors. BioNano has been great for that in some of my big projects. If Hi-C has been great for that. Also, the expectation is, if a new assembler is being developed, you're going to apply it to a well-understood sample like human. If you see, there are big problems there, the expectation is whatever rate of errors you see in a genome like human will carry over to other mammals. The dirty secret of this is, FALCON, which I was part of, GNU, MiniSM, all the long read assemblers do make mistakes. As we looked at different projects, maybe one big mistake every few megabases, every 10 megabases, there'll be one big mistake where you'll get, some sort of chimeric or inversion or something. So it's really hard to assess accuracy, but the best thing you can do is bring in different types of data to make sure that they all are in agreement. Okay. Thanks, everybody.